Hello, everyone. I would like to start by thanking the organizers for allowing me to tell you about my work today, looking at how noisy transcription controls stochastic cell fate specification in the fly eye. During development, immature cells will go through a series of transitions, or decisions, to become a fully differentiated cell of a particular fate. In most cases, we can think of these cells as making informed decisions through things such as signaling or lineage that can push a cell towards one fate over another. However, another mechanism to determine cell fate is through stochastic mechanisms. And so although these cells may receive cues from signaling and lineage, some cells may reach a point in which they randomly differentiate into a terminal cell fate. We use the fly retina as a system to study stochastic fate choice. The R7 photoreceptor cells express one of two photopigments, either rhodopsin 4 or rhodopsin 3, in a random pattern, as you can see here. This binary decision is controlled by the transcription factor Spineless. In R7 cells that express Spineless, they take on the rhodopsin 4 fate, while in R7 cells that lack Spineless, they take on the rhodopsin 3 fate. This results in the mosaic pattern of the adult retina, in which about 67% of R7 cells express Spineless and rhodopsin 4. But how is stochastic Spineless expression controlled? In order to understand this terminal cell fate decision, we must first understand how Spineless is expressed during development. The fly eye develops from the eye antennal imaginal disc in the larval stages. We take a look at a model of the developing eye during this stage. We see that the anterior region develops into the future antenna, while the posterior region develops into the future eye. In this model, Spineless expression is shown in green. Spineless is strongly expressed in the antenna, which we use as a positive control for all of our experiments. The eye, however, has a dynamic temporal pattern of expression corresponding to different points in R7 differentiation. The eye disc develops temporally from the posterior to the anterior. I will zoom in on this box of cells and walk you through Spineless expression during the different points in differentiation. All of these cells begin in an undifferentiated state where Spineless is off. Development begins at this posterior tip, and cells go through a precursor state where Spineless is expressed. We call this expression the early pulse. As precursor R7s continue to differentiate, they turn Spineless off. Finally, as these cells take on their terminal R7 fate, a subset randomly turns Spineless back on, while the rest remain off. These decisions then stay for the lifetime of the cell. This image leaves us with a snapshot in time in which all stages of development can be seen. Work by Katie Anderson and Luke Vortman in the lab has shown that Spineless is regulated by two enhancer elements. An early enhancer located upstream of the gene drives expression in precursor R7s, while a late enhancer located within an intron drives expression in the terminally differentiated R7 cells. We know that this early expression is required for this late terminal expression. And interestingly, if we modulate early expression, it alters the late on-off frequency. For example, if we look in a wild-type eye, we see RNA expression of Spineless in the precursor R7s, and we see that about 65% of the adult cells express Spineless. If we delete the early enhancer, we lose expression in the precursor state and subsequently lose expression in the adult. In a mutant of the early enhancer, we see a reduction in the number of the precursor cells expressing Spineless. This is then reflected by a reduction in the number of cells expressing Spineless in the adult. Finally, if we look at a deletion of a silencer element, we see an increase in expression in the precursor state and an increase in expression in the adult. But what I haven't told you yet is that in the antenna, and in the terminal R7 cells, we see expression of both RNA and protein. But in the early pulse, we only ever see RNA expression, never Spineless protein. We predict then that transcription is acting as a source of variability in these precursor R7s. For example, we predict that individual precursors express Spineless at different levels. This expression state is then retained as these cells differentiate. Cells then that had high expression in the precursor state will become on cells in their terminal state, while cells that had low expression early will become an off cell late. To test this hypothesis, I've developed an assay to measure transcriptional variability. I'm doing this using a multicolor RNA fluorescent in situ hybridization approach. The nascent transcript of Spineless is 30 kb in length, and so I've broken this down into three 10 kb chunks that I can label with different colored probes. As transcription progresses across this locus, we then expect to capture cells in different points of expression. Cells that are just initiating transcription will be a small red dot, while cells that have made full transcripts will co-express all three colors, as you can see here. In the antenna, where Spineless is constitutively expressed, we see co-expression of the 5' middle and 3' probe in each of these cells, supporting that Spineless is making full transcripts. Whereas in the early pulse, we see cells in different states of transcription. This cell here is co-expressing all three regions of the transcript. This cell is expressing only the 5' and middle region, while this cell is terminating expression and expressing just the 3' probe. And so qualitatively, we see that this variability exists, but what is the nature of it? I want to first emphasize that transcription is not always a continuous process as I have depicted here, and instead transcription can occur in bursts. And so keeping these different states in mind, I begin to measure the nature of this variability by comparing the fluorescence intensity ratio of different regions of the transcript. The output of this comparison is a distribution of ratios. This is an example of an antenna distribution. At the peak here, we see that the 5' and 3' ends of the transcript are expressed at about the same level in these cells, suggesting a continuous or steady state. Cells that shift to the right have a greater 5' to 3' ratio, while cells that shift to the left have a greater 3' to 5' ratio. Deviation in distribution in either direction suggests non-continuous transcription within those cells. We predict to see a spread in the distribution of the precursors, suggesting more variability. And so if we first compare the 5' and middle regions of the transcript, we see a narrow distribution in the antenna with a broad distribution in the pulse, seeing these cells shift to the right and to the left. Similarly, if we compare the 5' and 3' regions, we see this broad distribution in the pulse compared to the antenna. And finally, comparing the middle and 3' regions, we see exactly the same thing. This leads us to conclude that spineless expression is more variable in the pulse than it is in the antenna. But so far, we've only compared 2 regions at a time, and we want to know within an individual cell, how do all 3 regions compare? And so there are 6 possible relationships that we can see denoted on the left here. For example, this top relationship shows that the 5' end is greater than the middle, which is greater than the 3'. If we then classify each individual cell into one of these 6 categories, and we take a look at the distribution in the antenna, we see that most cells fall into these first 2 categories with the higher 5' intensity. This suggests that these cells are continuously initiating transcription and making more transcripts. If we look in the pulse, though, we see a distribution that is more spread across these 6 groups. Perhaps this spread is a result of more bursty or non-continuous transcription, giving us these different states, supporting that there's more variability in the early pulse. And so now that we have seen that this expression is more variable, we want to know if this noise can be explained by developmental timing. And so if you remember back to the development of this tissue, it occurs in a wave. And so every cell goes through each transition, but at different times. Cells in the anterior region are developmentally younger and less differentiated than cells in the posterior region. Therefore, as cells go through their precursor R7 stage and express spineless, the cells that are closest to the furrow here are just initiating this pulse, while the cells in the posterior have been expressing spineless for a longer period of time. If we then look at the RNA expression of spineless in the early pulse and consider this timing, these cells have been expressing longer than these cells that have just initiated expression. Therefore, it's possible that the variation we see is a result of this developmental timing and not of transcription itself. To address this, we fit the position of each cell in the pulse with a nonlinear regression. We then measure the distance of each cell to the center of the pulse, with the oldest and youngest cells being furthest from the center. If we then plot the fluorescence intensity of these cells as a function of distance from the center of the pulse, we would predict that if developmental timing does play a role, a relationship somewhat like this, with the youngest cells just initiating and having the lowest fluorescence intensity, and the oldest cells being on the longest, having the highest intensity. Alternatively, if developmental timing does not play a role, we would expect to see no relationship between fluorescence intensity and the distance from the center of the pulse. If we take a look at the 5' probe intensities, we see that there seems to be no relationship between intensity and position. This is recapitulated for all regions of the transcript, suggesting that developmental timing does not explain the variability in transcriptional noise. And so if we come back to this original hypothesis, we predict that the role of this noise or variability in early expression is to determine the late on-off state of a cell. And so for the next part of my talk, I would like to tell you how I'm looking to link this early state to the terminal expression state. And the way that I'm doing this is making use of the MS2-MCP system to monitor endogenous spineless expression. And the way that this works is you can incorporate this MS2 sequence into your gene of interest. As your gene is transcribed, these stem loops get incorporated into your transcript. These are recognized by a protein called MCP, which is bound with a fluorophore. And so in your cells that are on, you'll see a bright puncta at the nascent site of transcription, whereas in the off cells, you'll see an absence of this. And so I first had to get this MS2 system working in the larval eye disc, and we needed to be able to culture this eye and image it for 24 hours. And so I've optimized this system using a MS2 driver that is expressed in all terminally differentiated photoreceptor cells. And so at this point in development, you see these cells in this posterior region are expressing the driver and are terminally differentiated. We see the wave of differentiation move in this anterior direction, and as these cells take on their terminal fate, they begin to turn this marker on. And so we were able to successfully culture this eye for 24 hours and image this system. And so now that we have an assay, we want to know what aspects of transcription contribute to the noise in the early pulse. And I've created 4 different lines of flies to do this, each that contain insertions of MS2 stem loops into the endogenous gene locus at different positions. I have insertions in the 5' UTR and the first intron to measure initiation, the third intron to measure elongation, and the 3' UTR to measure termination. Using these lines, we see high expression in the antenna. And similarly, if we look in the early pulse and zoom in on these cells, we see expression of spineless in the precursor cells, recapitulating our RNA FISH results. And so we next want to know what happens if we compare these different regions of the transcript. And so if we take a look in the antenna, we see that most of these cells seem to express spineless as we would expect. When we quantify this, we see there's no difference in the number of cells expressing spineless with the first 3 insertions and a slight decrease at the 3' end, which is expected since these transcripts are terminating and leaving the nascent site of transcription. However, when we compare expression of each insertion in the precursor cells, we see something different. So in the 5' UTR, everything seems to look the same as our RNA FISH results. So does our intron 1 insertion. So we see expression in all of these precursor cells. But when we look at our third intron, we see a reduction in the number of cells expressing spineless, and in the 3' UTR, an even further reduction to only a couple cells expressing these. And so qualitatively, we see that the 5' end is expressed in more cells than the 3' end, with each insertion further from the start decreasing in expression. And so what does this mean for the source of variability? Well, we predict that transcription elongation may play a role in regulating the noise that we're seeing in the early pulse. And so keeping in mind that we only ever see RNA expression in these precursor cells, what we notice is that these first 2 insertions are located upstream of the late enhancer. And these insertions are expressed in more cells than these downstream insertions after this late enhancer. We know that this late enhancer is required for terminal R7 expression, and so perhaps transcription through this enhancer is variable. Without the need for the protein in these precursor cells, it's possible that transcription does not finish in some cells or is blocked and terminates early, potentially providing us with the variation necessary for this binary stochastic fate choice in the terminal R7 cells. And so what I've shown you today is that spineless is expressed variably in the early pulse. Developmental timing does not seem to explain this variation, and we predict that the source of variability may be elongation. The next steps are to monitor endogenous spineless expression from the precursor state through the terminal cell fate. And so this is the first ever movie I've made of endogenous spineless expression in the antenna using this MS2 system. And I was able to get this right before we went into shutdown for quarantine. And so we are working to now segment and track the transcription in each of these individual cells. We then hope to monitor the variability in each individual precursor cell and track that cell to its terminal cell fate to see if it becomes an on or off cell. And with that, I would like to thank everyone in the Johnston Lab, especially Luke Vortman. We work very closely together on spineless regulation, and you can check out all of the other work by the Johnston Lab at SDB this weekend. And if you have any questions, I'm happy to answer them.